right, welcome back to Facilitate TV. I'm joined by Rob and Felix from Excellos. Gentlemen, can you tell me a bit more about what your company does?
Sure, I'll go first. We are Excellos. We are a CDMO, but our novel approach is that we also emphasize the need for donor characterization. In other words, to make a better final product to treat the patient, you need to know that your starting material also is of high quality. And so we've developed assays and bioanalytical systems that allow us to characterize the starting material to assure better quality material. A key aspect of that, though, is monitoring in process the initial characterization, making sure that the phenotypic fidelity that you want of the cells does not vary as you go through the manufacturing process. And that's where the Excellos platform is very helpful, Felix.
Yeah, thank you. Thank you for having me. So the Excellos platform is a portable flow cytometer. And basically what we try to develop is systems that can meet the requirements of the manufacturers of CAR-T products or cell therapy products, but also bring the technology to the places where things happens, right? So in the context of the characterization of cellular materials, for example, we want to have a real data to support the characterization of these donors that Rob was mentioned, right? So to have a fidelity of the immunophenotyping and the characteristic of the sample before going into different other processes like cryopreservation or selection or others.
Right, and this would be a fairly large issue inside of the space, right? That we don't have proper characterization of the donor material.
Absolutely, and I think that Rob can talk more about that, but we are seeing some differences in the manufacturing process to success or to failure based on the nature of this starting cellular material. So the industry is start really paying attention and taking care of that this particular aspect. Say we need to have a better control in these leukapheresis materials, what we have inside, what is the nature of the cell, what is the balance between the CD3, CD4, CD8, etc. And Excellos push that reflection more far away than simply having an instrument that provide that information.
Very good. Yeah.
Right. So, I mean, one of the critical aspects of the Excellos platform is its portability means we can actually put it into the clean room. Now, typically, if you need to do in-process monitoring for a FACS analysis to make sure you're maintaining the right phenotype, the right CD4 and CD8 ratios, you would take a sample out of a biosafety cabinet in the clean room. You would then put it into a vial. You would then transfer it to an airlock. Okay, and on the other side of the airlock, someone would wait for the airlock to purge, open it up, decontaminate it, take the sample, walk down the hall to the FACS analyzer, and do the analysis and then report back to you. Whereas the Excellos device can sit in the clean room, 2 or 3 drops on the device, and it gives you the results in a matter of minutes. So far less risk in terms of manufacturing, time loss in terms of contamination risk and things like that. Because of that, when we first saw the platform, we bought 2 of them. And my thinking was we'd have one out in the main lab for analytical purposes, and we'd have one in the clean room as needed for in-process monitoring as it's appropriate for the different clients that we serve.
Gotcha. So give me an example here. The material is put onto the device. You get a reading. What's a decision or what's a next step that can be made based off of that information? So
actually, before it's even put on the device, let's say you do that part. Let's say you need to have a certain CD4 to CD8 ratio. So you put it on the device. The Excellos platform itself it's a ready-made cassette that'll give you the CD4 to CD8 counts in real time. And you know if you're there or not, and if you're not, if you need to make adjustments upstream to make sure you maintain the ratio, you can do so. But even before that, where Excellos, our company, comes in, is we characterize the donor starting material. So one thing we do know about cell therapeutics now is garbage in, garbage out.
Yep.
If you start with low quality products, why would you expect to get a high quality end product at the beginning? So we actually thought about that and we said, well, what does it take for an immune cell to cure cancer? And we focused on those attributes, not everything under the sun, because there's a million different data points you could collect. And we tend to do that as scientists. We get lost in the woods of all the new technology platforms that can generate mountains of data,
Right.
which by and large, most of that data does not have what it really needs to be useful data. By that meaning, it correlates with clinical outcome. So what we did at Excellos, we said, there's a million points of light in terms of data opportunities, but what data do we now know for sure correlates with a response that's a clinical benefit versus a response that is not a clinical benefit? What data sets matter? And the ones that we found were effector function, immune memory function, and metabolic fitness. If cells are not metabolically fit, or if they don't have immune memory, or if they don't have effector function, you will not cure the cancer with a T-cell based therapeutic.
Right.
So we focused on that data set as an initial assessment of the actual donor themselves. Now the donor can be an allogeneic donor for allogeneic products, or it can be the autologous patient themselves. One of the questions that's always come up is why don't CAR T cells cure all autologous patients? It only cures some of them. We now are more and more starting to realize that's because the immune cells that were used to make the product from the autologous patient were defective.
Right.
Now, why are they defective? It could be genetics, it could be age related, it could be therapy related, and that's a real scary one, because a lot of the times before you get a CAR T therapy, first you go through these massive chemo radiation therapy protocols that cause long-term damage to the immune cells in the patient. And then after you've done all that to them and it didn't work, then you say, well, now you're a CAR T candidate. So now we're going to collect those immune cells that we've already damaged,
Right.
and we're going to use them to make a CAR T product. Where's the logic in that?
Yeah.
But that's what we do now. Now, there's a light at the end of the tunnel. We're pushing to get CAR T therapies to frontline. Once we get them to frontline, then you will be collecting right up front before the cells have been damaged.
Definitely.
We're actually lobbying the community to say, why don't you go ahead and collect ahead of time now? Bank it. You know this patient's likely to become a CAR T candidate because of the type of disease. If we can predict, you know what, we're gonna give you standard of care because ethically we're required to do so, but we're pretty sure you've got a 90% probability that it's not going to work good enough for you and you're going to become a CAR T patient. If you already know that, why not preemptively collect the immune cells before you damage them? Bank them, have us test them to make sure they're good cells, and use those to make the product. We're actually doing studies on that right now at Excellos in collaboration with other groups. And we'll be using the Excellos platform possibly to do some analysis on the FACS profiling as well. So it's exciting times because we're hoping to improve the therapeutic outcome for the patients this way.
Definitely. Yeah. Wow. Anything to add there?
Nothing, you know. So no, what we realize is obviously the community needed new tools, new innovations, right? So flow cytometry has been there for years, but the regular flow is not meeting the expectations the manufacturers have today. So in our company, we only trying to miniaturize the flow cytometry and making simple, because we have proprietary technologies to write down reagents, to make also auto-classification algorithms, so there is no bias in the way the data are analyzed, right? So bringing more standardization, and we hear a lot about standardization of the process, but it's also in the analytics that needs to be standardized to compare that site A and site B are making the same thing, right? So and this is what we are trying to do in collaboration with Excellos too.
Gotcha.
And that's the other part of donor characterization for allogeneic products is one allogeneic donor may have this level of potency, another may have this level of potency. The quickest way in the world to confound your clinical trial results and make your clinical trials last longer and become more expensive is by having variability in the potency of your final product. That is a cardinal sin. Right now, it's interesting in that in the cell therapy space, we're okay with that. We say, well, it is what it is. We get what we get. We don't agree with that. At Excellos and Excelix, we believe there's ways to standardize starting material qualities and properties, only pick high-performing donors that are similar, and use that and know that year in and year out, you're making a similar potency product month to month or year to year for your 5-year clinical trial that's going to cost you hundreds of millions of dollars to run.
Right.
It reduces the risk of failure. It also reduces the risk of higher variability in clinical response, which then requires you to power the study with a lot more patients and a lot more time and a lot more money. And even then, you may not reach the threshold of a statistically significant result to win. So we're trying, with the approaches that we use here, we're trying to better harmonize and standardize to a higher degree of quality the products that are used to treat the patients. The hope being that we see far more cure, far more successful clinical trials, and by doing that and improving the potency on a per cell basis, we lower the costs of goods. If you lower the cost, how do you do that? Well, if you've got a more potent product, you give a lower dose for the same efficacy. Does a couple of things. One, it lowers cost of goods. Two, it makes it lowers because you give a smaller dose, but it also improves safety because you're giving less of the therapeutic product with the safety liabilities it has in terms of cytokine storm. So we're excited. It's a new era again. Now the basic concept of CAR T and TIL therapies is out there, and there's been approvals. We know that it can work, but we also know there's plenty of room for optimization still to go on to make a better product for the patient.
Makes sense. So I would love to understand in a bit more detail, right? If I am just spinning my cell therapy out of a lab versus when I'm just before my IND versus when I'm now into clinical trials, where are you guys able to come in and help there at these different stages?
So we can, both with the Excellos platform for profiling whether or not the donors or the patients have the right profile, immune profile of CD3 T cells and CD4 and CD8s. We also can then tell you, are these cells able to kill? Will they proliferate? Real big question. Our algorithm actually will tell you whether or not the cells will proliferate or not. Our algorithm will tell you whether or not the cells will kill or not in a cell killing assay. Our algorithm will tell you whether or not the cells have durability. In other words, metabolic fitness is a critical piece. We're now realizing more and more in the clinic that the metabolic state of the cells is critical to their ability to kill, but also to be durable and hang around. Because if you have a very good CAR T product, but it dies off after the first round of kill, you still got 50% of the tumor or more there that you haven't addressed yet.
Right.
You need them to be serial killers. That requires stamina, cytological stamina. That goes to metabolic fitness. And the analogy I would have is, let's say we both have the effector function. I have a 50-pound strap backpack of rockets and bombs on my back to vanquish the enemy, and you do too. Okay, if the enemy's sitting right next to me, we can both equally destroy it.
Yeah.
But if the enemy is in a distant part of the body, which is analogous to it being a half mile away up a 20-story building, and I'm told I have 15 minutes to get up there and kill it, and you have 15 minutes to get up there and kill it, I'm never going to get there. However,
I might not either.
Well, but we do know that age recapitulates.
Right.
So it's interesting because we've done studies showing that metabolic fitness, as you get older, decreases on a cellular level, not just on a whole organism level. So you've got a better shot at getting up there than I do. So if I have my choices, I'm going to use you as my therapy, not me.
Right.
Because I can do the stuff right here, but I can't do the stuff over there. That's where metabolism is critical.
And I think that is also another point is, so all the technologies we are developing is helping from an operational standpoint, right?
Right.
So and all the work they are doing is to be more predictive and anticipate risks rather than document the failures.
Right.
Right? Because the failures is the worst case that we may expect, right? Patients are behind, waiting for the products, and this could be very costly too. Right. So all this together, the analysis of the donors plus the new analytics that we're bringing to the market can make a big difference.
Very good. All right, so in a nutshell, 30 seconds or so, in a nutshell, what is the eScore platform and how can people use it?
The eScore platform is composed of 3 primary factors, actually 5. Age, of course, a BMI, body mass index, effector function potential, which is a proteomics-based screen checking the cells to see if they have the weapons to kill, metabolic fitness, which is basically looking at glycolysis, which is a key aspect that cells have to go through to kill, metabolic fitness, and then memory, immune memory. If the cells can't remember, 80% of all tumors that kill people because of recurrent disease, not the primary tumor. We can get rid of the primary. It's the recurrent metastatic disease that kills 80% of the time. That's where immune memory is required. Without immune memory, you get recurrent disease and the patient can succumb to it, or you have to give them another round of treatment. But if you have robust immune memory, that will be those residual cells that remain in the autologous patients in particular will cure. In fact, Emily Whitehead,
Yes.
she still has immune memory.
Yeah.
That's why she's cured.
Indeed.
Yeah, absolutely.
Phenomenal. Yeah, so really looking at those different components that are going to impact the ability for that therapy to last long term. Makes a lot of sense.
Yeah.
Thank you.
Gentlemen, perfect. Thank you so much for your time.
Thank you.
Thank you. Thank you for having us.